Behavioral Neuroscience, Chapter 17, Segment 4. Donald Hebb proposed that when a presynaptic neuron repeatedly activates a postsynaptic neuron, the synaptic connection between them will become stronger and more stable. A cell assembly is a large group of cells that tend to be active at the same time because they have been activated simultaneously or in close succession in the past. A Hebbian synapse is a synapse that is strengthened when it successfully drives the postsynaptic cell. Researchers placed electrodes within a rat hippocampus, stimulated presynaptic axons, and recorded the response of postsynaptic neurons. Low-level activation of presynaptic cells produced stable and predictable EPSPs. When they applied a tetanus, an intense volley of action potentials, to presynaptic neurons, the response of the postsynaptic neurons changed. The synapses now appeared to be stronger and more effective. Long-term potentiation is a stable and enduring increase in the effectiveness of synapses following repeated strong stimulation. This hints at a cellular basis of memory. The hippocampal formation consists of the interlocking hippocampus and dentate gyrus, a strip of gray matter, and the hippocampal cortex. Three major divisions within the hippocampus are labeled CA1, CA2, and CA3. In CA1, LTP occurs at synapses that use glutamate and is critically dependent on NMDA receptors. There are drugs that can block NMDA receptors and prevent new LTP in the CA1 region, but they do not affect LTP that has already been established. The release of glutamate in low-level activity in the CA1 regions activates only AMPA receptors. The NMDA receptors cannot respond to the glutamate because magnesium ions block the NMDA receptors' calcium channels, allowing less calcium ions to enter the neuron. If larger quantities of glutamate are released, AMPA receptors respond more strongly. Since they emit sodium ions when activated, this increased activation depolarizes the postsynaptic membrane. The magnesium ions are driven from the central channels of the NMDA receptors, allowing them to now respond to glutamate. The large influx of CA2+ at NMDA receptors activates protein kinases, enzymes that add phosphate groups to protein molecules, causing a functional change in the proteins. One is calcium MK2, which causes more AMPA receptors to be produced and inserted into the postsynaptic membrane. The net effect is to enhance the sensitivity of the synapse to glutamate. CREB is a CAMP-responsive element-binding protein that binds the promoter region of several genes involved in neuroplasticity when activated by CAMP. CREB causes genes to change their rate of expression and is activated by protein kinases. Inhibition of protein synthesis prevents longer-lasting LTP. Neurons can make proteins that selectively inhibit CREB's actions, possibly providing a means to erase or inhibit the formation of unwanted memories. When the postsynaptic cell is strongly stimulated, it releases a retrograde messenger, a transmitter that is released by the postsynaptic region, travels back across the synapse, and alters the functioning of the presynaptic neuron. The retrograde messenger ensures that more glutamate will be released into the synapse than previously, thereby strengthening the synapse. LTP can occur without NMDA receptor activity in other parts of the hippocampus, and some forms of LTP are blocked by drugs with completely different forms of action. The time course of LTP bears strong similarity to the time course of memory formation. Pharmacological treatments that interfere with LTP tend to impair learning. Training an animal in a memory task can induce LTP in the brain. This is known as behavioral LTP. Adult neurogenesis occurs primarily in the dentate gyrus. These new neurons play a role in forming new memories. Newly generated neurons in the dentate gyrus are more plastic and show enhanced LTP. Rats given a drug that is lethal to newly born neurons are reportedly impaired on test of conditioning when there is an interval between the CS and US. This shows that the role of neurogenesis in memory may be limited to hippocampus-dependent forms of memory. A conditional knockout is a gene that can be selectively deactivated in specific tissues and/or at a specific stage of development. In mice, this resulted in a marked impairment in spatial learning with little effect on other behaviors. Activation of newly generated neurons in the olfactory bulb enhances olfactory learning and memory. Working memory and the ability to form new episodic and declarative memories usually decline with age, but autobiographical memory and semantic knowledge tend to remain stable. Beta amyloid is a protein accumulated in patients with Alzheimer's disease. Older people show less cortical activation than younger people when encoding or retrieval is self-initiated. Age-related memory impairment may involve steady loss of synapses and neurons in the hippocampus and cortex. Memory impairment in Alzheimer's disease seems to be caused by the loss of cholinergic inputs to the hippocampus and cortex from the septal complex and NBM. Drug treatments that enhance ACH transmission improve aspects of memory performance in human participants. Nootropics are a class of drugs that enhance cognitive function. Ampakines act via glutamate receptors to improve hippocampal LTP. There is evidence that in humans, physical activity, mental activity, and adequate sleep can postpone cognitive decline in old age. Studies show that these factors can reduce cognitive decline as we age. Living in favorable environmental circumstances, involvement in complex and intellectually stimulating activities, and having a spouse or partner with high cognitive status. Fear-conditioned mice became afraid in a location they did not normally fear when neurons in the DG responsible for their fear were stimulated with blue light.